Hello, Dr. Hoài. So today our group would like to present about the T lymphocytes. These are the members of our group, Trâm Anh, Bảo Chiêu, Đức Dương, and Minh Thư. So our topic will include the morphology, function, and the application of T lymphocytes. So now first I would like to talk about the morphology of T lymphocytes. So these are some of the characteristics which are shown on the slide. T lymphocyte is one of the main adaptive immune system, and they recognize antigen through clonally expressed antigen receptor. And T cells are produced in the thymus, and they are require antigen to be processed and presented to them by specialized APCs. So there are two types of morphological lymphocytes. The first type is relatively small. It is typically granular and has a high NC ratio. The second type is larger, has a lower NC ratio, contains cytoplasm, isophoric granules, and it is known as the large granular lymphocyte. So T cell can be distinguished by the different antigen receptor. The definitive T cell lineage marker is the T cell antigen receptor, which is called TCR. The two defined T cell TCR are the heterodimer of two disulfide linked polypeptides, alpha and beta chains, and the second one is a structurally similar heterodimer consisting of gamma and sigma polypeptides. Both receptors are associated with a set of 5 polypeptides, which is called the CD3 complex, and together form the TCR complex. Approximately about 90 to 95% of blood T cells are the alpha beta T cell, and the remaining 5% are the gamma T cells. So next, there are two major subpopulations of the T cells. Alpha beta T cells are subdivided into two distinct non-overlapping populations. One subset carry the CD4 marker and mainly help or induce immune response, which is called the T helper, and the other subset carry the CD8 marker, the CD8 plus T cell, and is predominantly cytotoxic, which is called as a TC. And CD4 plus T cell recognize their specific antigen in association with MHC class II molecules, whereas the CD8 T cell recognize the antigen in association with the MHC class I molecules. So that's all for the two main types of the T cell. Here is some of the other type of T lymphocyte. First is the suppressor, which is also called the DREX. The suppressor T cell express the biomarker CD4, FOXP3, and CD25. Because effector T cells are also express the CD4 and CD25, tracks are very difficult to effectively discern from effector CD4+, making them difficult to study. Next type is the memory T cells. There have been numerous subpopulation of memory T cells suggested. The single unifying them for all memory T cell subtypes is that they are long-lived and they can quickly expand to large number of effector T cells upon their re-exposure to their cognate antigen. By this mechanism, they provide the immune system with memory against previously encountered pathogens. Memory T cell may be either CD4+ or CD8+, and usually express the CD45RO. And the last type is the NKT, which is called natural killer T cells. NKT cell is expressed a restricted reptile of TCR arrangements, and that are selected via the MHC-like molecule CD1D. NKT cells include both NKT1 plus, NKT1 minus, as the CD4 plus, CD4 minus, CD8 plus, CD8 minus cells. Natural killer T cell can share other features with NK cells as well, such as the CD16 and CD56 expression. NKT cell may initiate T cell response. NKT cell have T markers and also some the NK cell markers. So this is just the morphology of some kind of the T cell. Next part, my friend will introduce to you the function of these types of T cells.
My name is Chieu, and now I will present the next part, functions of T lymphocyte. The first one, helper T cells. This kind of cell will secrete the cytokine to activate other immune cells toward up the invader cells and do not interact directly with the antigen. Interleukin-2 is one of example of cytokine. It will stimulate the proliferation of helper T cells, cytotoxic T cell, and B cells. B cell will be differentiated into two kinds. The first is plasma cell and B memory cell. The stimulate the plasma cell development means that produce more antibody to enhance the ability to resist against pathogen, and it also enhance the activity of natural killer T cell. The mechanism of activity of this cell is similar to the NK cell in innate immune system. Helper T cell also secrete B cell growth factor to enhance the antibody secreting ability of the activated B clone, some chemical to attract neutrophil and macrophage to the invader to phagocytosis. Macrophage migration inhibition factor to inhibit the macrophage from leaving the area means that less macrophage stay in the infected area and activated eosinophil and promote IgE development parasitic infection. Now the second kind of T cell is cytotoxic T cell, or also known as CD8 or killer T cell. The cytotoxic T cell will secrete buffering a chemical substance to form a pore on the surface of invaded cells to the fluid in the cytoplasm penetrate into the cell and make the cell swell and burst, and also the enzyme delivery. This kind is an enzyme protease that lies the protein in the cell to make the cell apoptosis is a programmed cell death and lethal to tissue cell invaded by virus. And now suppressor T cell or regulatory T cell or Treg in short prevent autoimmunity. Regular T cell along with helper T cells. Immune tolerance. It means that modular the immune system maintain the tolerance to cell antigen and prevent autoimmune disease, general suppress induction and proliferation of infected T cell. Suppressor T cell will spread the activity of helper T cell, B cell, and macrophage. And the last one, memory T cell, are activated by antigen but do not enter in the circulation, remain in lymphoid organs, migrate to the various lymphoid T cells throughout the body. If attacked by same antigen again, immediate immune response, more powerful response in the next time.
Hello everyone, my name is Ann. I will continue my group presentation with the application. The first application that our group want to mention is chimeric antigen receptor CAR T cell therapy in cancer. To enhance T cells' ability to identify cancer cells, CAR receptor can be expressed on the surface of T cell, providing them with a specific cancer targeting mechanism. First, what is chimeric antigen receptors? Its abbreviation is CARs. It is engineered receptors. It is consists three parts. First, CAR specificity is frequently determined by a single chain variable fragment composed of an antigen binding region typically derived from monoclonal antibody. The second part is transmembrane domain that tethers the antibody to the cell, and the last part is intracellular signaling domain CD3 zeta contains co-stimulatory molecules such as CD28 and tumor necrosis factor receptors such as OX40 and CD40L. This is enable a more efficient and long-lasting activation of T cells, but often tumors do not express appropriate ligand for such co-stimulatory molecules. So to improve the responses with incorporate the additional secondary generation CARs such as CD28 and CD137. Next is the mechanism. There are 5 steps in the mechanism. First, extracting cells. T cells are collected via apheresis, a process that withdraws blood from the body and removes one or more components, such as plasma, platelets, or white blood cells. The remaining blood is returned back into your body. The second step is reprogramming. A viral vector inserts into T cells the genes carrying the instructions for CAR. And the third step is manufacturing. To elicit a powerful response in patients, we need to return many more reprogrammed T cells than they threw out. So CAR T cells are expanded in a bioreactor with the help of magnetic bits coated with 2 antibodies, anti-CD3 and anti-CD28, that signal T cells to proliferate. After expansion, which takes out 10 days, the magnetic bits are washed out. The fourth step is patient preparation. The patient is given chemotherapy to lower white blood cell count, thus increasing the immune system will accept the modified T cells. And the last step is treatment. The reprogrammed T cells are infused back into the patient's blood. Once circulation, they search for and destroy the cancer cells expressing antigen targeted by CAR. Next is the advantages and the limitation of CARs. It does not require antigen processing or MHC restricted presentation. HLA independent antigen recognition, and it's engineered to target 40 molecules such as protein, carbohydrate, and glycolipids. The limitation of CAR is it can generally only detect the cell surface tumor-associated antigens. This is the end of my part, and my friend Thư will continue our group presentation.
My name is Thư, and now we'll talk about the second application, which is the role of T regulatory cell in the development and prevention of type 1 diabetes. So normally in a healthy person, the pancreas produce beta cells that make insulin to uptake glucose in the blood and deliver them into the cell. While in the person with type 1 diabetes, the immune cell may have some mistake and fail to recognize its cell, so it will attack the pancreatic beta cell, which is called the autoimmune cell destruction. This leads to the absolute insulin deficiency and require lifelong insulin treatment. However, insulin treatment is not acute and it cannot prevent possibility of serious effect from type 1 diabetes. Therefore, scientists are looking for other long-term management options. In many experimental models for type 1 diabetes, immunotolerance was obtained following treatment with immunosuppression. In most of the cases, the immunotolerance relied on the expansion of Treg rather than on the deletion of energy of effector T cells. As my friend had mentioned above, the regulatory T cell is formerly known as the suppressor T cell, a subpopulation of T cells immunosuppressive that will suppress or downregulate the induction and proliferation of effector T cells. Therefore, they can modulate the immune system, maintain tolerance to self-antigen, and prevent autoimmune disease. And
the Treg will express three important biomarkers: the CD4, CD25 on the surface, and the FOXP3. The FOXP3 is the most important factor for the Treg, as the presence of this protein is necessary for their development. The Treg are generally classified into two categories: the natural Treg and Treg, and the adaptive or induced Treg, iTreg. Even though they have some distinguished features, like the natural Tregs primarily emerge from the thymus, whereas the iTreg are generated in the peripheral from naive T cells after antigen exposure. Post T cell subset share a similar phenotype that they express the intracellular transcription factor FOXP3 and secrete the interleukin 10 TGF-beta. The Treg will affect the effector T cell by four main pathways. So in the picture here, the first pathway is suppression by inhibitory cytokine. This pathway includes the secretion of three factors: the interleukin 10, interleukin 33, and the TGF-beta. These three factors will inhibit the secretion of cytokine in the T effector. The second pathway is suppression by cytolysis. The Treg will secrete the granzyme A, granzyme B dependent, and the perforin dependent killing mechanism to break down the T effector. The third pathway is suppression by metabolic disruption. This includes the CD25 to mediate the apoptosis of T effect, the cyclic AMB to mediate the inhibition, and the CD39, CD73 to mediate the immunosuppression of the T effect. The last pathway of the Treg mechanism is the suppression by targeting dendritic cells. This pathway includes the mechanism that modulate the dendritic cell maturation and function, such as the lymphocyte activation gene 3 on the MSC class 2 to mediate the suppression of CD maturation and cytotoxic T lymphocyte antigen, the CTLA4, CD80, and CD86 mediated induction, which is an immunosuppressive molecule. So it will stop the function of, inhibit the function of CD and the effector cell, T effector. So so using this mechanism, the treatment with the Treg will restore the function of pancreas to produce insulin normally and prevent or stop the development of type 1 diabetes. So it also bring me to the end of our group presentation. Here are some reference we have used. So thank you for your listening.